Hello, so my name is Chris. Thank you for tuning in. So today I'm just going to be, as the title says, I'm just going to be explaining to you what I did in lab today. And today was just a very technical day, very assay oriented. What I'm doing first, as kind of like a background, I'm taking bone marrow out of mice, and then I'm stimulating it and differentiating it pretty much with different factors to eventually get it to become a different cell type. So either macrophages or dendritic cells, which is what I did today. So the way we started off first was, and I'm going to be doing experiments with these macrophages and with the dendritic cells, and I'm going to eventually take their RNA, which I can explain at the end of the video, but I'm going to take their RNA, isolate it, purify it. Then I'm going to go and analyze it on qPCR for a set of different primers for certain genes that we think are upregulated. So we're looking at the mRNA transcriptional level by converting it to cDNA. I'll explain this later. So today what I did. So you have a mouse. So we have mice right here, and I took 2 mice that were wild type, abbreviated as WT, and then I also took 2 mice that were axle knockout. Pretty much, how do I want to put this? Axle is important to know in my lab. It's one of the 3 receptors of this family called TAMs, T-A-M. And in the TAMs, we have tyro3, axle, and MER. All of these are phagocytic receptors. They help with the clearance of apoptotic cells for ephagocytosis, but that's more conceptual based. So we have these 2 mice. We have 4 mice total, 2 from each. So what we're going to do first is, after sacrifice, you draw the mouse out onto a metal platform kind of like this, and you extend its legs. So you push pin all these areas in. And what you're doing, and here's the tail, and here are the ears. So what we're doing is we're going to be harvesting bone marrow from the femur and tibia. So that's these bones right here. So we go through this process after sacrificing, and we get one stubby bone like this and a longer bone like this. Femur and tibia. So then from each mouse we get 4 bones. From each genotype we have 8 bones total. So now I have my 8 bones, and what happened was, so when you're harvesting the bones from them, you're stripping away all the tissue and stuff, so you just get the bone, and inside the bone is bone marrow. So that's what we want. But, so what we're doing is when I'm taking out the bones, I'm putting it into sterile PBS, cold, and I'm keeping it in there while I'm doing this. This process can take a long time depending on how well you trim away the skin and the tissue prior to using the bone scissors and getting the bones out. But that's a different topic. So we have 8 bones from each mouse now. So now what I'm doing now, or jumping forward, is now I'm trying to clean all the bone marrow out. And I think in a previous video I talked about this, but what we do is, and this is a very generic description, you have a bone kind of looking like this, and you're gonna cut off these areas. So now that you're gonna only have this central region. Okay. And in the central region, you're going to put a syringe. And you're going to shoot, I don't know, however much you want into it, but you're going to flush out all the bone marrow, and you're going to hold all the bone marrow together from all the wild type. So I'm going to have, so I'm going to have all the bone marrow from my 8 bones from the 2 mice, and I'm going to save it in like a specific dish. So then we have this. So this is the process we're clearing it out with. You can use PBS. You can mix the PBS with DMEM or whatever. Like, doesn't really matter. But you have this process to get the bone marrow. So now, after that long process, we have 2 plates. Pen switch. So we have 2 plates, and cool. So we have 2 plates, and they have our 8. We have bone marrow from 8 bones in each one, and they're wild type and axle knockout. Okay, so that's cool. Now what we're doing from here is, it's not really in a dish like this. We put it into like a falcon tube, and like 50 milliliter falcon tube is fine. And so this is a mixture of bone marrow, progenitor cells. Well, pretty much it's all the bone marrow, which has like other stuff in it as well, and it has PBS, which is what we use to wash. So now we have it all in like this solution, like homogeneous, heterogeneous actually. But what we need to do now is we need to spin this down in order to separate the bone marrow from the PBS. So after we spin it down, you'll get a nice pellet at the bottom. And you'll have like the rest of your PBS, which doesn't go to the bottom because the bone marrow is way heavier than the PBS. So it gets down here, and this, I don't know, the spin time, you're looking at like 500 G for like 8 minutes. Something like this. And this is like very conserved, and we use this over and over again. So you're to this level, and so now you separate this out. Now you're going to aspirate, which is just like suck up all the PBS. So now you actually only have the bone marrow left. So you now have 8 bones worth of bone marrow at the bottom of your falcon tube. And we have 2 falcon tubes because 1 falcon tube for the wild type, 1 falcon tube for the axle knockout. But we need to now use ACK lysis buffer, which then pretty much kills off a lot of the cells except for the bone marrow progenitor cells. So we're going to add ACK lysis buffer. Usually you can add like 5 milliliters per mouse So you add 5 milliliters of ACK lysis buffer per mouse into this. So we have now a test tube, or now a falcon tube with only the bone marrow in it, but now we're going to add 10 mLs of ACK because we started off with 2 mice. So we now have a solution of these bone marrow with 10 mLs of this ACK. Then you wait like 1 minute or whatever, and then you're going to add in 2 volumes worth of some like DMEM or something like that to neutralize the ACK. So you're going to have now 2 volumes, where 1 volume is 10 mLs. So you're going to add 20 milliliters of CDMEM, complete DMEM. And so the total volume is now 30 milliliters. So now we have 2 tubes with 30 milliliters of like either CDMEM and ACK lysis buffer. And right now, the pellet when we first spun it down was red. So erythrocytes were in there, blood cells were in there. So that was kind of bad because we don't want them. They don't differentiate for us. So we now mix these 2 together. So now our solution is just like it was before with the PBS mixed in with the bone marrow. So now what we're doing is we're going to spin again at this same like parameter, and now we're going to go and have another look at our falcon tube. And it's going to be now a pellet at the bottom, but the pellet's going to be white. because there's no more erythrocytes, because you already aspirated out all the ACK and all the CDMEM. So now we have all the now we have all that pure bone marrow progenitor cells. So we're excited. So the next step that we're doing from here, which I'll erase all this stuff, is now we have the bone marrow progenitor cells. Now what we have to do is we have to count them. We have to see exactly how many cells were, or how many bone marrow progenitor cells we have. So this is done like via light microscope, like a regular table microscope, and pretty like kind of like this. But so you have a solution right here. So each mouse. So these are C57 black 6. So just to say that it's black 6 mice. Each black 6 mouse has 4 bones and will roughly yield you like 30 to 34 million cells. That's usually what it is. So we should be expecting like 64, maybe 70 million cells from the 2 mice. So we know that it's going to be 70 million cells, or something around this. So there's these counting grids, which looks kind of like this, and then there's like a section in the middle, a place for your pipette, and then like a small like grid, which you can't really see until like, you look under a microscope, but it looks something very similar to this. So yeah, it looks something kind of like this. And what you're going to do is, I'll show you, I'll label the squares that we count. So we count these major group squares. But so you have like, so your goal is to get like, I don't know, 100 counts per square. And if you, and one square will then yield you, so the average of these 4 squares will give you the total of the number of cells times 10 to the 4 per milliliter. So if you get 10, if you count 10 cells in here, then you're going to have 10,000 cells per milliliter, because this average is like times 10 to the 4 cells per milliliter. So you can't see this very well, but you can hear what I'm saying. So we have to try and think of what a good concentration would be to get it so that we're around 100, assuming that we know that this is going to be roughly around 70 million. So like for me, like what I did today was I resuspended in like 5 milliliters. So I did whatever solution that I wanted. So like for macrophages, you use macrophage media, and the macrophage media has like a whole bunch of like different factors in there. One of them is like L929. But so you're going to have 5 milliliters in here. So we have a 5 ml solution with our pure bone marrow progenitor cells, BMPCs. And now what I did was I did a 20x dilution, because like if this, I were to count this whole square would be like white with all these like dots, and you couldn't really count it very well. So I did a 20x dilution of my 5 milliliters. So I took out 50 microliters of my solution, and then I resuspended it in 950 microliters of like my CDMEM, which means that this is like 1 in 20. Because if you imagine like 100 and 900 would have been 1 in 10 dilution, because you do like, because it's like 50 over 950 plus 50, which is 50 over 1,000, which is 20x dilution, because you would need 50 times 20 would be would make it so that it works. So, so you count it on this plate, and we're just making sure that we have the right number. And then when we figure out we get the right number, we then would calculate it out to like what the number is supposed to be. So we do this times our dilution factor times the number of milliliters, roughly, and we get like the total number of cells. So what you can do is you can spin down your 5 milliliters again, aspirate it all off, then add the media back in, add some new media back in, so that you get a certain cell concentration. 1 million cells per milliliter, 2 million, 3 million, 6 million, whatever you want, that's what you're going to do. And from there, you're going to now plate that on your 6 well plates, 15 centimeter well plate, 15 centimeter dish. And so you're going to plate it on there. So that's pretty much what I did today. I plated one 15 centimeter well dish per mouse. So these 15 centimeter well dishes, I plated them, one for axle, one for wild type. And then beyond this, I did a conglomerate of 15 wells on a 6 well plate, which means that it was like, so for each genotype, I used 2.5 dishes, where each dish had 3 mLs in it, and yeah. So that's pretty much what I did today, just that kind of in a nutshell. Hope this helped, hope it was interesting. Yeah, thank you for tuning in.